Okay, so welcome to the Sea AMA. This is the second episode of the Sea AMA. Last week, if you tuned in, you would have heard myself and Dora and Milan talking about the sea projects in general, talking about the token, about the dApps, about Seastarter and all the other great stuff. Yeah, and the point of these AMAs is to share important information, really, about projects, about issues, about important ocean projects. It's to give a platform to individuals and organisations working in marine science and other environmental fields. So that's how it's going to be moving forward. We're going to have people on here talking about a whole range of different stuff in marine biology and beyond. With that in mind, this week, we're extremely excited to have with us on the AMA series Kristina Loosen, who is the brain behind our first Seastarter supported project. So before we get started, just a very quick intro. If you are not familiar with Sea projects, I'm James, I'm the founder of Sea, and I'm in Scotland on the west coast. Sea is a blockchain project designed to address a number of different problems in ocean science and marine biology. We're building dApps to help us to gather environmental data on blockchain in a more efficient way and a more accessible way. We launched Seastarter, which is a platform to support new environmental projects. It's like a Kickstarter kind of thing. And we support 8 huge environmental organizations, including Sea Shepherd, Oceanic Preservation Society, Coral Reef Alliance, and several others. So that's us, and that's me. And also in the AMA with us today is Dora, our project manager. Over to you.
Hi everyone, I'm Dora. I'm helping develop our project, and yeah, I have 20 years of experience in the crypto and tech sphere. I've worked for Google and other educational tech companies. Yeah, and I'm excited to be here.
Awesome, thanks, Dora. And yeah, so without going on about ourselves anymore, I would like to introduce Kristina Loosen. Kristina, could you tell us a bit about yourself and tell us about the project?
Yeah, sure. Hi, everyone. Thanks for having me. And so I'm Kristina, and I am a marine biologist, and I am lucky to be the first one to be supported by the Seastarter project. And so I've studied in my bachelor's. I actually studied biological sciences, so I wasn't specialized yet in marine bio, but I always had a dream to be a marine biologist, so that was always the plan. And for my master's, then I went to South Africa, where I started to work with sharks, which are my absolute favorite, which you might have guessed already. So since my master's, I've been working in different jobs. I was an entertainment supervisor in an aquarium, and I was a local sea turtle biologist for the Olafritti project in the Maldives. And now I'm finally moving back to the shark topic because I'm about to start my PhD in South Africa again, and I will be investigating the assessment tool eDNA to analyze the species distribution of sharks around the South African coast. And I'll be supported by Seastarter and Sea, which is amazing because that's the only way we can actually realize this project. We are very happy to have that opportunity, definitely. So yeah, that's just a quick wrap up. I'm actually, maybe one important part is I'm originally from Germany, which is where I'm located at right now. But I'll be moving back to South Africa to start the PhD in October time, hopefully. So yeah.
Awesome. Thanks, Kristina. And I mean, we're going to get into the project in some detail, I guess, now, because we've got some amazing questions from Twitter. And I dare say there'll be more questions from the chat and stuff like that. But yeah, thanks so much to everyone who has submitted questions in advance on Twitter. Some really, really good questions there. So let's get into them. We got a question from Mr. E. And the question is, why did you choose sharks? Are they more responsive to climate change than other marine animals? Which is a great question.
Very good question, definitely. So as I mentioned already, it was always my dream to research sharks. So I definitely try to find a project which is studying sharks, which is asking questions about sharks which haven't been answered yet. And in this case, with the eDNA project, and the question behind it is, what is actually happening? Where are the sharks moving to? And this is also in relation with climate change, because as we all know, has been a lot of stuff going on in the world's oceans, and it is affecting lots of animals. And sharks are top predators, which we call apex predators. So they are on the top of the food chain, and therefore they are very specialized often. So they are very sensitive to changes. So if something in the food chain changes, they will feel it and they will react to it. So they're kind of like a beacon for what is going on. If something's happening to the sharks, it might be happening on the lower levels of the food chain already in way worse conditions. And also often sharks are, as they are the apex predators, they are often also key species in an ecosystem, which means they are crucial to the systematics of the ecosystem. What is actually going on? They keep the entire glue together. They're the glue that holds it all together. That's what they often are. So if something's happening to them, the whole system might collapse. So this is why sharks are extremely interesting to study, but at the same time very difficult to study, because as you might imagine, it's very difficult to monitor the entire ocean. It's very wide and it's very deep, so it's very difficult to actually find these sharks, to know what is going on. And this is where eDNA comes into play. But I think there's another question on that topic as well. So I'm going to be talking about the detail of how eDNA actually works.
Brilliant. Thanks for that, Kristina. Well, yeah, that is actually the next question from Natasha Lois is, can you explain how the shark eDNA works? So what devices do you use to find out what species of sharks have passed in the ocean, and how reliable is it?
Okay, that is actually a very, very good question, because this is what we also try to find out as well, because eDNA, which stands for environmental DNA, is a fairly new method. So what we will do is very simple, actually. It's very simplistic. So what we do is, there is in every environment, be it terrestrial, be it marine, or be it freshwater environment, there's animals, there's plants, and everything is shedding DNA material, be it skin or saliva or what other particles of an animal's body. So they leave behind genetic traces, and with the eDNA we try to sample that and then analyze it. and thereby we can tell which animals have been around. So in our case, we will pretty much just need a bucket of water, and in that water there we hope to find DNA fragments of all kinds of sharks. So that is the first step. So very, very easy. So literally you need a bucket of water, you take it out of the ocean, you can go to different depths, or you just take it from the surface, and then you cool it, and then you have to filter that water. So all that water, of course, needs to go away, and we just want the DNA material in the end. So we filter it through a normal, custom, whatever water pump that you can buy in every store. And we filter it through a specific DNA filter. And on that filter, we will have the DNA material which was soluted in the water. So we take that and then we freeze it until we can further process it. And the next step is something which I guess everyone is kind of familiar by now, because the next step is we need to extract that DNA, and then we have to do a PCR test, kind of test. So PCR has been probably one of the words of the year 2020 and 2021, but probably not everyone knows what it actually stands for. It is, so PCR means polymerase chain reaction. And the thing, what it does, it's also very easy when you think about it. So when we have this tiny DNA fragment from a shark, it's not readable. We can't just see that little piece of DNA. So what we need to do is we have to multiply it. We have to multiply it and multiply it and multiply it. And this is what a polymerase chain reaction does. It takes that fragment, it reads it, and then it also duplicates it. And then it duplicates it again and again, and thereby it's an exponential growth of that specific DNA fragment. And thereby we will be able to tell which species it is. We then have to further sequence it, but that's another step. But actually, this is pretty much the whole idea of it. So with COVID, it's the same idea. We have a little bit of COVID virus, then we multiply it in the PCR reaction, and thereby we can tell, okay, there is the virus. Beforehand, we wouldn't be able to tell. And this is the same thing with the DNA. And what we also need is we need a specific primer. This is like the start tag for the DNA that we want to read, because in that sample, of course, there's all kinds of DNA from fish, from marine mammals, from seabirds, and we don't really need that. We don't really want that. So we need to have a specific primer which attaches to a piece of DNA which all sharks have. So then just the shark DNA will be duplicated and multiplied, and then we can just tell the shark DNA and the shark species that we have there. That is pretty much the idea. What was the last part of the question?
It was how reliable is it?
Ooh. Okay, true. So yeah, we actually don't really know yet. It's, as I said in the beginning, it's fairly new. So in scientific terms, fairly new means it started to be used more regularly at around 2012, which is very recent. And but there has been a huge increase in studies using eDNA because it is a really nice method, but still we are not entirely sure how reliable it is, what we can actually take from it. So there needs to be more studies to analyze exactly that question. So we can't really say yet. So this is also why I'm doing this PhD. I want to know, can we really use that?
Brilliant. Yeah. So this is brand new stuff. This is really really groundbreaking stuff. Yeah. I mean, yeah, 2012 is, in terms of scientific discovery, that's a brand new start date for technology.
Yeah.
Yeah, I mean, the next question we had is from Kosti, and it's in the same vein. They asked, how accurate the eDNA sample test is, which we've kind of covered, but also I guess comparing it to how it used to be done.
Mm-hmm.
So Kosti asks, how much time do these tests take compared to the old ways of doing things? And what were those old methods? And you know, how can these tests and studies help shark species and other species as well? I guess that's a separate question. But,
yeah. It actually helps me to just explain the whole idea. So the questions are really good to start from what is it, how we use it, and why we're using it exactly. So that is perfect. The first question was, what were the old methods? The old methods were, for example, we took, or we are still taking, there are still lots of studies that use the old techniques. We take DNA samples by taking a skin sample or a muscle sample, or we can also use blood samples as well. So thereby we would have to catch a shark and then take a little piece of muscle tissue and then release it again, which is, of course, a little bit invasive. There's a lot of stress involved in that method, but we have a definite DNA sample from that individual. Then also what is a little bit less stressful is photo identification. This is when we have people seeing the sharks. There's a visual cue. So we see the shark, and then also the people have to be certain what species it is. So there is quite an error to that. But with photo identification, you can also trace individuals, you can trace populations by that, but you always have to see the animal, which, as I said in the beginning, is a little bit difficult with the sharks because they tend to be quite deep, or they be underwater, and we as humans still can't hold our breath for so long. And diving is also restricted to a certain depth and to a certain amount of time. So it's just not very easy to monitor them. And then also what we can do is with the diving itself, so we can go underwater and try to find the sharks. But most of these methods are either invasive or expensive. So it takes a lot of time, a lot of effort to find the sharks. Then we can tag them as well, of course, so we can monitor one individual's movement, which is definitely, like, this gives us all a lot of information, but it is quite expensive as well. So there is some downsides to the old ways, but of course there's also still some downsides to the eDNA, which we try to find out and which we try to maybe eliminate. But one of the downsides of eDNA is that so far we can't really be certain about the amount of DNA. We can't be certain about the specific specimen, so we can't really know the individual. There has been one study done on whale sharks where they have sequenced the DNA fragments down to the individual whale sharks that they saw. So it is possible, but still tricky, and so there is still lots of stuff that we need to do. So that is about the old techniques, and the advantages of eDNA being it doesn't cost as much. Like the sampling itself, it's so easy. As I said, you need a bucket of water, a water pump, and that's about it. Of course, the processing of the sample later with the PCR, you need specific chemicals, you need solutions. So they cost, of course, something, but in the end, it's still way cheaper than lots of equipment and all that stuff. So that is definitely one of the advantages. And what was the last part of the question? There's always so many questions packed in one that I tend to forget the last part.
The last part is, I mean, I guess this is tied to a lot of people have asked similar questions. It was, how can such studies help shark species and other species?
Yeah, that is, of course, we need to know why we're actually researching things, right? There always has to be an outcome, a higher purpose to that, because it doesn't really help us as much if we just look around and, oh, well, that's interesting, but so we want to, of course, always improve the situation of sharks. They are, as I said in the beginning, they are apex predators. They are crucial to most ecosystems. They are, as many people know, like, it's just the common things that are said. They are kind of like the garbage cans of the ocean. They eat everything that is old and weak, so they clean out the oceans and keep it healthy. They prevent diseases. They keep populations in check. So this is all very important. And the baseline for that is we need to know where they are, how many there are, what species are there, to know where we need to protect them, how we can protect them. This is like the baseline of what we need to properly improve conservation efforts. And with South Africa, it has been a little bit difficult lately, because there has been quite a huge shift in certain shark species distributions, especially the white shark. There has been quite a huge change in the ecosystem. Might have heard about it, that since 2016 or 2015-16, there has been less and less sightings of white sharks in common aggregation sites, one of them being the Seal Island in False Bay, or one of them being Gansbaai, which is one of the white shark hotspots. And in the recent years, there has been less and less sightings. And even some of the time in the high season, there have been no sightings at all. And we are still not entirely sure what causes that. There is different hypotheses which might cause that, but we are not sure. And therefore, with an eDNA sample, we might be able to tell where they are. And then we can go on for the question why they're there. But we still first need to know where they are, what species, and then we think ahead and try to figure out why they're there, or where they went entirely.
Yeah, absolutely. That's a really good answer.
I've heard that we have no idea where they give birth.
Yes, true.
Crazy.
We have still no idea. There is some areas, one of them being off the west coast of Australia, and one other one being off the west coast of Northern America in the Baja California, which is where they believe to be a white shark nursery. But still not entirely sure. But this is where we often see juvenile young of the year sharks. Young of the year means they've been born in that previous year. So this is the areas where we think that they might have given birth, but we have never seen it. We don't know anything about it. Actually, it's just lots of guessing and hoping to see it at one time. Actually, also interesting, even the Mediterranean is suggested to be a nursery area, which lots of people tend to forget, that there used to be a lot of great white sharks in the Mediterranean. By now, there are so few that it is like finding the needle in the haystack. So, but it used to be totally common to have great whites in the Mediterranean.
So these are the kinds of mysteries that eDNA can solve for us, hopefully.
Yes, exactly.
Wow.
It could be a very, very helpful tool, and I really hope that it will become more and more common to use it. There's lots of futuristic thinking about what we could use it for. It's really interesting. So, but I don't want to give it away. I don't want to spoil it. it might never happen, we don't know, but there's some really interesting ideas.
Amazing. I love it, by the way, when you say that we as humans still can't breathe underwater for very long. It's like I can hear the frustration in your voice. Like
I've been trying. I've been training my free diving. I've been practicing, but I still can't access 4 minutes. But well,
yeah, we need to
just want to become a fish. Yeah.
Yeah, we need to evolve.
If someone finds a scientific way to do that, I'll be the guinea pig, definitely.
Oh my God.
Brilliant.
I think Milan will join as well. I think he would also totally be down for that.
Yeah, for sure, definitely. Cool. Let's move on to some more questions. We've got a couple more to get through. I think some of it we've already covered, really. Well, I guess one interesting question that came in was from George Liam, and he was asking about, I guess he's asking about documenting how you go.
Is James breaking off, or is it me? James? Is he still there? Dora, I also can't hear you.
Frozen.
But you're there.
Let's just continue with questions that we received on Twitch until he reconnects. So
Sorry about that. I lost my
all right.
That's
this is the problem about living in the ocean. Sorry.
I was about to say you might reconsider living on an island.
Definitely. Yeah, the question was about how do you plan to document things? Will there be any video and stuff that we can watch?
Definitely. Yeah, I'm planning on keeping everyone up to date as much as possible. Of course, my supervisor, Sarah Andriotti, would tell me that my focus should be on the PhD, but I also want to share it, of course, with everyone. So yeah, I'll once I get down to South Africa, and even before that a little bit, once I have finalized all my planning and my travels, then I will try to keep everyone up to date. I will definitely try to stream from the boat, but I can imagine that the connection is almost as bad as in Scotland, so we'll see. But I'll try to document everything, and I definitely want to share some beautiful white shark or shark videos with you. I'm so excited to see them again, so I definitely want to share that joy with you as well. Because,
how nice. Keep posting images and new findings.
Yeah, with the findings, we need to be we can't be too upfront about it. We can't share it before we haven't statistically analyzed them. So of course we need to keep that. Like, I share everything that's going on, but the results need to be statistically absolutely waterproof. So yeah, but yeah, everything else, and I can always give you little snippets and stuff.
Awesome.
I also plan.
Sorry, go on. Carry on.
I was saying I also plan to join a conference, and then I can also take you guys along there. Conferences are always very, very interesting to meet other shark scientists. It's lovely. For me, it's kind of like going to the concert of my favorite band, because I get to meet researchers that I've been reading for years now, and it's kind of like meeting my idols, and so it's always lots of fun, and you get to talk about sharks all the time, which is, as you might imagine, my favorite thing to do. And so yeah, I'll take you guys along on that as well. I'll share the beautiful nature of South Africa with you. I just can't wait to get there.
Yeah, we will definitely join you on that. I think that's the end of the Twitter questions. So thanks so much for everyone that sent those in. Dora, you've been gathering live questions while we've been
Yes.
on the air.
So one idea, I think Sea Token should create a mobile game working with token and contributing to the project and also raising awareness and education. What do you think?
A game.
A game, yeah.
Okay.
I think that's a pretty nice idea. I think, I think actually it's the kind of thing that we would support through Seastarter. If somebody fancied coming up with an educational ocean game, maybe whoever sent that question in, then have a think about it.
Head to seastarter.org.
Yeah, it's definitely
someone who has programming skills. Definitely go ahead. I might not be the one to do that.
You can consult them.
I can, exactly.
The educational part.
I'm definitely here for that.
Yes. So another question is related to that. How do people like Kristina get in touch with Seastarter? Is the Seastarter advertised somewhere?
Yeah, definitely. Seastarter.org. You can get in touch with us straight on there, and we're right there. We're ready to talk to you about your ideas and your projects, and
we haven't advertised it as in ads, but we have sent it out to hundreds of journalists and hundreds of environmental activists. You can maybe shed a bit of light on that, James.
Yeah, I mean, Seastarter is gradually becoming quite big news in sort of environmental circles. We will be talking about that a bit more next week on the AMA, actually, with the We Naturalists crew. I'll tell you more about that in a bit. But the word is gradually getting out there, and we do get a lot of applications, but that shouldn't discourage you from applying if you have a good idea. So.
A question: what is Kristina's favorite type of shark?
That's always my favorite question because it's the easiest one to answer always. Since I was about 10, 11 years old, I don't know why or how, but I got the idea in my head that I wanted to be a marine biologist. And the main reason for that was because I was watching ocean documentaries at the time on German TV. And I was sitting down, actually taking notes while watching the documentaries. And the white sharks were just the most amazing animals that I've ever seen. And it has been the same since then. White sharks are my absolute favorite animal in the world. They are so beautiful. And up until I was, let me think about it, I think 21 or 22, I have never seen a white shark, neither another shark in the ocean myself. So I've never seen a shark live besides an aquarium. So this is why I went to South Africa to try and test if I actually like them, if I actually enjoy seeing them in the water, or if I might maybe even be afraid. You know, like I had that idea in my head, but it could have gone totally wrong because I might have been there in the water on the boat and might have been scared to death. But luckily that was not the case. So I went to South Africa to test that theory. And so when I saw my first white shark, it was, and still is, the most amazing moment of my life. They are such beautiful animals. They're so graceful, and the sheer power and the majestic aura that they have is just...
they have some kind of crazy electromagnetic sensory organs, right? Can you tell us a bit more about that?
That is not just the white sharks, it's actually all types of sharks. And they have these gel-filled, pores on the head and on the snout, and they are called the ampullae of Lorenzi, and they can feel electromagnetic, or how you say, electromagnetic movements or beats. They can feel that. So they can feel your heartbeat, they can feel your movement, not very far, so it would have to be fairly close to the prey animal or to whatever they've been investigating. So that is one of the additional senses. They are so cool.
Wow.
Another question.
Can we I think this is a really good point, just to maybe talk about that a little bit. You said about maybe being scared of the sharks and you didn't know if you were. I mean, this is a big thing, right? People are scared of sharks, and most people have never actually encountered a shark, but they're scared of them. Why is that? Why are we scared of sharks?
It is actually, unfortunately, it's leading directly into a primeval fear. Sharks, they play into the fear of big teeth, definitely, and they also play into the fear of darkness and depth, which is from the ocean deep. So this is what Steven Spielberg played with very well in the well-known movie Jaws, it just stuck with people, and because gotta say, like, the movie is done very well, so it actually works very well with getting people very scared of sharks. Funnily enough, Steven Spielberg and the writer of the novel that it's based on, the author, they both are totally biggest shark advocates by now. So they try to reverse what they have kind of like introduced, or kind of like started with the Jaws and the shark fear. So they try to conserve sharks as much as possible. And with the scared, I gotta say, Jaws has been a major factor in it. But of course, there has been people scared of sharks before that. And it's just been distributed in the media more and more. So I think everyone thinks about, maybe I might be afraid of sharks. And I wasn't sure, because I loved watching them, but it might have been a totally different case. And that is also probably related to the media attention that they get, or the coverage and the way they are portrayed. That is definitely also why I even questioned myself, even though I love the animals. So being scared of sharks is a combination of primeval fears and a combination of the media. And I always try to point out that I do not dismiss being scared of sharks, but I dismiss being totally freaked out by sharks, thinking they are man-eaters, they are just beasts that eat everything. So this is what I dismiss. I don't want anyone to feel that way. I can understand why people might be scared, but it needs to be a rational fear. So they are predators, and depending on the size of a predator, they are potentially harmful to people. This is just the way it is. The thing is also with sharks, there is very small species. Most of all shark species don't get bigger than 1.5 meters. So they are not really potentially harmful to humans. Of course, there is some other ones which are big enough to cause major injuries, and therefore, of course, they can be dangerous. But they are not mindless eaters and monsters. Every time I saw them, they were beautiful. And I've interacted with lots of people on boats. For example, I was working on a cage diving boat in South Africa, in Cape Town. I was working with shark explorers, and I was helping the guests with everything. And there were some people which got on the boat and they said they were scared of sharks. So they were facing their fears. And every single one of them got out of that cage smiling from ear to ear. They were so happy and they were so amazed by the movement, by the beauty of the sharks, which is, you can just see that when you see them in real life, because unfortunately on the media they still portray always it's like the wide mouth with the big teeth. It's rarely just the shark swimming along calmly and peacefully, and this is what you get to experience when you see them in real life. So every one of these people, they totally reversed their view of sharks. It was so amazing to see. So this is what I want everyone to understand, that I can understand if you're scared of them. Just don't be completely frightened and irrationally scared of them. Just keep your senses together. Don't be stupid around them. And that's the gist of it. Of course, there's more to it, but
yeah, it's a great message, definitely. Do we have any more questions, Dora?
Yes. Kristina, do you guys use the new eDNA technology already, or is it still being developed?
Being what developed?
Bought or developed?
It's not. It's an ongoing process. So there is not one definite eDNA method. There is different protocols. So there is different ways of sampling. There's different volumes that you can sample. So you can either take a small milliliter sample, or you can take a liter of water. It depends on the filters that you use. It depends on the solutions that you use, on the primers. So it depends on every individual study and what your question might be. So there is a development always ongoing, and it will be the same way in the future as well. So every individual researcher and study has to think about, what do we want to show, what do we want to test, and thereby you have to change the protocol accordingly.
Yeah, so people are discussing, do they actually think we're food, or?
Well, unfortunately, I can't look into a shark's head yet. I don't know what they're thinking. But no, it's a difficult question. The thing is, people tend to humanize animals. So I can't really say what they think, if they think at all. There are predators. They are relying on instinct a lot. They are relying on their senses. And what I always like to point out is that the shark has lots of very good senses. As you said earlier, with the electromagnetic sense as well. They have also a perfect, for example, white sharks, they have a very good sense of smell. They can hear very well. So they are very well equipped to scan their environment. So I would never think that they mistake us for food. I would always suggest it's rather an individual accident, or it's just an individual animal which is just more curious. But I wouldn't think they see it as food. It's just with sharks, they're mostly opportunistic. So if given the chance, they might also try human for a moment, which is of course very tragic. And depending on the shark, it can be of course lethal. But there's always the fact to point out that there is, on average, there's 7 to 8 people who die through shark-human incidences, which is a low number, of course, as I said, very tragic. but it is a very, very low chance for that to happen. So I wouldn't think they see us as food, because think about it, how many people are in the ocean every day. And if you've ever been to South Africa, people go into the water where they know there are white sharks and they're common around there, and people still go into the water. And there is, of course, safety measures implemented. There is a system in place, especially around Cape Town, where there is shark spotters on every mountaintop, scanning the shores for sharks. And if a shark is sighted, then depending on the shark's activity, the water might be evacuated. So everyone has to go to the beach and wait for the shark to move some other area. And this is a very safe method and non-lethal method to keep humans and sharks safe. And still, I was there at one time when there was a shark sighting. So I just arrived to go surfing at Muizenberg Beach. And there was a shark sighted, and everyone was evacuated from the water. Everyone waited until the shark had passed onto another area, and then we all went back into the water. No one even thought about it twice. So if they would be a mindless human killing machines, no one would be surviving going into the ocean at all, because they're perfect predators. So yeah, we couldn't defend ourselves against them.
What if when they start really starving?
It could, of course, influence the reaction of a shark towards a human. But from my experience, most sharks are very, very cautious. They are they are also vulnerable. They try to keep themselves safe as well. They have some vulnerable areas like their gills and their eyes. So when they prey, they always close their eyes and they try to keep their gills away from teeth and whatever. So they are very cautious animals. So I really think that it happens that a shark just goes for the kill immediately. That is mainly talking about white sharks, because this is the species that I've been interacting the most with. There's other species like oceanic shark species. They are in the open ocean, spending most of their life there. And in the open ocean, there's not as much prey. So they are very opportunistic. So if there is prey around, they are more likely to go for it. So these type of species like the oceanic white tip, sometimes the mako shark, they might be a little bit more prone to going in for the bite a little bit more quicker than any other species.
Do we have time for more questions, James?
I think we've got time for like one more question. Have we got one?
Why not NFDC token to raise awareness for oceans and species?
Why not what?
NFDC token.
Well, I mean, NFT is obviously a very popular technology right now. It has a lot of different use cases. It has a lot of different possibilities. I think we're only just seeing the beginnings of what's possible with NFTs. And there are a lot of people saying to us, why don't you get involved with NFTs? I think everybody's getting involved in NFTs at the minute, and mostly not for the right reasons. So I think it's something that we're definitely interested in. But I think we would only get involved in it when there's a clear fit between what we're doing and NFT use. You know, like even the cryptocurrency C token, we're working in the field of cryptocurrency as a funding mechanism for seed projects, for our projects on blockchain. So, that is a clear use of cryptocurrency. NFT could work in the same way, but until there's a very clear link, until it's obvious that we should do this precise NFT project to help Sea grow, then we're interested, we're looking at it, and we'll step in definitely when there's a good moment.
It has some connection with helping oceans, right?
Absolutely. There's a lot of people out there, a lot of artists, a lot of musicians, writers, sound recordists, documentary makers. There are people making amazing media to do with the oceans. And all that stuff is a very rich bed of material for NFTs, obviously. So, yeah, we're thinking about it. Okay. Well, we're through the Twitter questions, we're through the live questions. I think we'll round it up at this point. Thanks so much, Kristina, for coming on. It's been really amazing. It's been a learning experience for all of us.
Pleasure. As you might think, I'm happy to talk more about sharks, and especially about the topic of are sharks dangerous? What are the different factors to it? So we might have another AMA about just that. And then you guys can ask me all your questions about sharks and shark-human incidences. And I'm happy to hopefully change your mind about sharks.
Brilliant. Yeah, we will definitely do that. We will definitely do that. So we had the Twitter contest for the questions. So let's say now the winners for that. We'll reply to that tweet so you all know who you are. But the winners will be Mr. E, Natasha Lois, Kosti, Put Lucky Ace, and Rimuru. So well done, guys. Brilliant questions. Thank you so much for sending those in. Right now in our Telegram, we're going to launch the quiz. So we're going to post the link to the quiz in a few minutes. Give us like 5 minutes after we close the AMA. We'll post the link to the quiz. Go in there and answer those questions. If you've been paying attention and taking notes, then they're easy. And yeah, there's rewards in there as well. Next week on the AMA, we've got a bunch of people who are running an amazing project. They've just started up this year, I think this year. It's a project called wenaturalists.com. And it's a kind of a social network for people who are involved in or passionate about nature. So it's a great way to sort of build relationships between conservationists, technologists, people like us, people working in the field, in all kinds of nature projects. Everything from, literally everything you can imagine in nature and conservation. So they're going to be on the AMA next week answering your questions about that. That's going to be really exciting. I think that just about does it. Thanks again, Kristina. It's been awesome. And thanks, Dora, for helping out again. And
thank you, Kristina.
Thank you, guys.
Yeah. We'll see you all very soon.
Bye, everyone.
Thanks, guys. Bye, everyone.